I can foresee a time where immunotherapy and CAR T therapy move to the forefront and replace chemotherapeutics as the standard in these solid tumors. So I'm Omid Hamid. I'm a medical oncologist at the Angeles Clinic and Research Institute. We are a Cedars-Sinai affiliate in Los Angeles. My role is the Director of Translational Research and Immuno-Oncology, and in that role, I work to bring new therapies to patients who need other options in order to fight their cancer. Translational medicine is taking what we've learned in the lab and then bringing it into the clinic, so from bench to bedside, and doing it in a fashion where it's the most available for our patients. The most exciting aspect of my job is the ability to work as a detective to understand a patient's tumor and its tumor tendencies and translate that into optimal therapy for them. I came into oncology initially because I was extremely happy with the proximity to patients, where you have a close relationship. I've stayed in oncology because of the science, and what I've seen is a revolution in immunology and a revolution in oncology coming together because we've been able to utilize now informatics and immunotherapeutics together to take patients and give them an optimal therapy. My focus is on trying to harness a patient's own immune system to be able to combat their cancer. And what we've learned early in other solid tumors has been able to bring us forward to utilizing similar therapies for gastric cancer, pancreatic cancer, and other gastrointestinal cancers. The standard at this point is to begin with chemotherapy. Unfortunately, the majority of patients do not have the benefit that we would like with chemotherapy. That's when immunotherapies and clinical trials come to work. We then can see if newer agents that work by a different mechanism can help those cancers. For example, immune checkpoint inhibitors, which has been a majority of my research to date, are drugs that come into a patient's system and relieve the immune suppression. They resolve the breaking of the immune system and allow the immune system to target these tumors. Early data has shown benefits in pancreatic cancer, in esophageal cancer, in gastric cancer, and other GI malignancies. Through that mechanism, we've understood that the immune system is extremely important in all solid tumors. Similar to chemotherapy, checkpoint inhibitors do not give you 100% benefit for all patients. And therefore, that's where a therapy like CAR T comes in, where you can offer a different mechanism to inciting the immune system and harnessing it to control these tumors. CAR T cell therapy is chimeric antigen receptor T cells. These are T cells where we have taken a patient's own T cells from their blood and then pheresed them or taken them out, and using a viral vector, like a virus, been able to insert a DNA sequence that codes for a receptor, something that targets a protein found on those tumors. The proteins that the CAR Ts target are not expressed in normal tissue, and therefore you don't get that attack on your normal tissues, but they're overexpressed on the cancer, the malignancy. So almost like making this T cell that has no direction a heat-seeking missile. These T cells that then go back into the patient's body, they traffic directly to where the tumor is, and they can have persistence. They can be there for an extended period of time. It has the ability to recognize that tumor and those proteins on the tumor as foreign, and then connect, making an immune synapse. And that just means that the T cell, our own immune system, and the tumor get together. the T cell becomes activated, exert its action on the tumor and cause tumor lysis, which is tumor breakdown. By causing that tumor to break down, it causes an immune cascade where the rest of the body can then be recruited and other T cells to control disease, not just in a local area, but throughout the patient's body. We have a lot of proof of principle of that in not solid tumors, but liquid tumors, like leukemias and lymphomas. We are now able to translate that work into gastric cancer. Currently, there are multiple CAR Ts that are targeting multiple proteins on gastric cancer, and there is a specific one that targets claudin that's had an FDA orphan designation, which means that there's enough excitement over it that the trials are ongoing to make this hopefully a standard. A cancer that has been so refractory to every therapy, like pancreatic cancer, has shown response and benefit to immunotherapy. We have now standards of care for esophageal cancer and gastric cancer that utilize immune checkpoint inhibitors. The future for gastrointestinal cancers is the understanding of our ability to now utilize multiple immunotherapeutic platforms, whether it be checkpoint inhibitors or CAR T cells, or a combination of both, along with other immune-targeted agents. These therapies are being utilized at earlier and earlier time points to prevent those situations that become life-threatening. The future of clinical trials in gastrointestinal oncology is multiple different immunotherapeutic drugs together.